Okay.
Hello, everyone. I'd like to welcome you to the NF California webinar, NF Studies and Clinical Trials in California. I'm Kim Bischoff, the Executive Director of the National Neurofibromatosis Network. The NF Network is the leading national organization advocating for federal funds for NF research and building support and community for NF. NF California is a member organization of the NF Network. Maria, Katie, and Debbie Bell and others from NF California are a part of the NF Network Advocacy Program, which brings together NF patient organizations from around the country coming together to speak as one voice on the importance of federal funding for NF research. The NF Network truly appreciates the work that NF California has done to prepare for this webinar this evening. I'd like to turn it over to you, Katie.
Thank you, Kim. I wanted to introduce Debbie Bell right here, and Maria in the royal blue, and myself, Katie Spearing. We work we interchange to try to work as one to make things like this possible for the NF community, because we all have someone with neurofibromatosis in our family. So what we wanted to do first was ask Dr. Maureen Boshian from UCI to give a presentation that kind of addresses the idea of considering joining a clinical study or trial. So Dr. Boshian, please go ahead.
Okay, let me just get this on the screen for a second. Okay, so we'll talk a little bit about clinical trials, about what they're all about how to find them, and about how to find some information that will help you decide about joining a research study. So basically, a clinical trial is an experiment or observation using human volunteers, because the researchers want to answer certain questions about either medical or behavioral treatments. And they may want to study a particular kind of patient, a particular disorder, a medication, and see which dose is the best, a new indication for an old medication, a new treatment to see if it's any better compared with a typical treatment. And they may be studying vaccines, as everybody knows, because of all the COVID studies, medicines, drugs, procedures, other kinds of things, medical devices. And they want to know how safe they are and how well they work. It's important to clarify when you're looking at a study that the study has been approved by an institutional review board or a similar board, which is generally composed of physicians, researchers, other healthcare professionals, community members, to make sure that the study is ethical, that the risks are as low as possible, and that the subjects, the people who participate in it, have their rights and their welfare and safety protected. If they approve the study, it doesn't mean that it's a safe therapy or an effective therapy. It's just saying that it's ethical and reasonable for the study to be conducted. And actually, when you look at these studies, if we're talking about drugs, only about 10% of all drugs that are studied in human trials, human clinical trials, become approved. Clinical trials, why do we have them? Because we usually don't know when there's something new, if it's going to be helpful, if it's going to do harm, if the risks versus the benefits are skewed in one direction or the other, or if it could be, no different than what's already available, including doing nothing compared with doing something that doesn't work. So we need to know if these things are safe, if they work, and to measure certain outcomes in the participants. For example, somebody may go into a trial for high blood pressure treatment, and they may get a drug or some other treatment, and then they want to see, the researchers want to see whether your blood pressure goes back to normal or not. That's a very simple kind of study. And they may compare this new approach to one that's already available, or to a fake drug, which is a placebo, an intentionally ineffective treatment basically sugar water or something like that, so that they can separate out the kinds of reactions that people may have just subconsciously, just because they know that they're getting something, either thinking that it's helping them or thinking that it's hurting them. But they have to make sure that that those kind of false impressions don't get mixed up with the real data. Who is responsible for them? There's generally somebody called a principal investigator. That is the study big shot. Often a medical doctor working together with other individuals from the health professions or social workers or other people that are important to get this to get the study right, including statisticians and others. The payment for conducting the study could be by a pharmaceutical company, it could be a university, the government, or specific individuals. And they can occur in a multitude of different places, hospitals, universities, community clinics. It depends on who's conducting the study and what needs to be done in the study. Just very quickly, I'm not going to go through all of this, but you will see as you look up trials that you may see that they're in specific phases, particularly for drugs. And what they do is they start out small and give a drug to a small group of people just to understand how it functions, how it works physiologically and in the body. And then if that comes out well, they'll give it to a larger group of people to make sure it's safe, to figure out what kind of a dose to use, to see what kind of side effects there are. And after that, they will go to another phase 2 trial, where they usually will have a treatment group compared with the fake drug group. And the people in getting the real treatment that they're studying or the fake treatment will not know what they're getting. So they can't be biased by knowing that they're getting either the treatment or the fake drug. Then they'll go to a much larger group of people, anywhere from 1,000 to 3,000 people, to get additional information. And once all of that passes, then the drug can go on the market, but then they keep collecting information as people are using the drug to make sure that there are no dangers and that they don't have to make any modifications or possibly even take it off the market. There's something called a research protocol, and that's the plan for the study. And that should explain to people the reason for the study, who is eligible and not eligible. And I'll show you an example of that, how many participants the study needs, what the schedule is, what they're going to do, what they will do to the volunteers who participate in that, how long the study will last, and what kind of information they need to get from the volunteers. And you must sign an informed consent, and informed means informed. So there will be a consent form that presumably has gone through the institutional review board that should be in clear language that anybody who's not medically adept can understand. The purpose is to protect people, to make sure that you absolutely understand every important thing about this study. And once you sign an informed consent, you are not obligated to anything. If you sign the consent and you start the study and you don't like it, you can leave at any time, even if it's not over, and no problems will be there for you. So you're not required to continue once you decide to start. The questions, the kinds of things you need to ask: why are they doing it? What's it about? What do I have to do? How long is it going to take? How much effort from me or traveling or whatever will it take? Are there going to be any risks to my health? Are there going to be any unpleasant side effects? What about the information that I give you to conduct the study? There may be information about me, my age, my weight, health situation. Who's going to have access to that, and how will my privacy be protected? How is this new stuff different from my usual stuff that I'm getting? For instance, if it was that blood pressure study, how is this new medication going to be different from the medicine I'm usually taking? Could this help me? Can I get some benefit out of this study? Are there any alternatives to participating? Is there something else that may be better for me to do than participating in the study? Could I get the same medication if I decide not to participate in your study? Could I get, will it cost me anything? Do I have to pay for treatments? Excuse me, for transportation? I'm sorry, my timer just disappeared, so that's going to be a problem for me. Anyway, will insurance cover the costs? If I'm injured by the study, will the study pay for it? Will anybody pay for it? Do I find out the results of any tests on me? Or do I ever find out the results of the study? And many times you cannot find out the results of tests because the lab is not an approved lab for doing clinical testing. So here are some clues, and I'm going to go through this relatively quickly, but I can give the sponsors of this webinar a PDF file of these. So people, if you want the URLs, you can get them. Probably the best place, in my opinion, to look for clinical trials is something called clinicaltrials.gov, and that's the URL also that you use. And when you go to this web page, you will come up with this page that's here, and it will be called Find a Study. So what you want to do is probably look at the ones that are recruiting, the ones that are looking for participants now, or the ones that are planning to but haven't started yet. If you look at all studies, then you'll get a bunch that have already closed, and that may be making it more difficult for you to find the ones that you might want to participate in. Then you'll enter the disorder that you want, and I entered neurofibromatosis, and then I got a pop-up menu of some subtypes, and you can either just stay with that, or you can pick another one that's more specific. Once you do that, and I've got that same page over here, then you put in where do you live, because this is international as well, even though it's a US government site. So I put down United States, California, Los Angeles, and within a 50-mile radius, and then it will, if you put search, it will look up the trials for you. And for NF on that, I got 11 studies that are open and recruiting, meaning they're looking for people. And you can put in all sorts of limitations on this. The one that's not yet recruiting but open and just recruiting, you can put in ages, you can put in sex, you can put in other different kinds of study types, and it will search on those bases. And I'm going to enlarge this one because I know it's tiny. So we'll just go to that. That one, the trial that I picked is a topical gel, some medication that you smear on your skin, basically for adults with NF1, and it's looking at the skin neurofibromas. And what it's telling you is here is the drug, that the name of it, and that there's a, the type, that it's a gel type of medication. And then it will tell you where the study sites are. So there are 3 in Los Angeles. No, I actually can't see this. So there's at least 1 in Los Angeles, but it'll show you a few, and then there are 16 other ones. So you could link on this and find out where there's something, a study site that would be close for you. Once you link on that particular study, then it will show you the title of the study, and a lot of information about what it's about, again, for skin type neurofibromas and what the medication is. And then it gives you the actual official title, which is too long to read through here, but it will explain basically what the study is in quick language. If you go farther down on the page, you will get, for this particular study, what ages are eligible for the study. So adults 18 and older, all sexes. It does not accept volunteers who don't have neurofibromatosis. It tells you what the inclusion criteria are, so give you a whole bunch of limitations on what you need to have in order to participate. And then it tells you the exclusion criteria, that tells you if you're doing this, and there's a whole list below this of what kinds of things would keep you out of the study. For example, it says if anybody has used any steroid creams or ointments on their neurofibromas or on their skin within the last 30 days, you don't get to participate because that could falsely affect the outcome of the study, either make it look better or make it look worse or whatever. So it will give you all of this information. So don't forget, this one probably is the first one that I would go to. But there are a number of other sites. This is at the NIH, the National Institutes of Health, the National Institute of Neurologic Disorders and Stroke. And it includes studies on neurofibromatosis, and you can look here at finding clinical trials and then look up which trials they have available. Then there are other sites. If you just do a Google search on clinical trials, this one, I looked up neurofibromatosis in some of these sites and really couldn't find anything going on. So again, they're not always available in these different web pages. One called Clinical Connection, another one called Boulder Science, and so on. And then you can find them sometimes in patient databases, newspaper advertisements, other places. You might find them on social media, other sites that might give you some information. This is an excellent site. It has information on why should I participate in a clinical trial. This is an NIH government site. It gives you the basic information in some, very good educational material about the basics of clinical trials themselves, stories of individuals who volunteered, finding clinical trials, and so on. There's another one that's very important. It's called the Office for Human Research Protections, and it will give you information about how a good clinical trial will protect your safety and your personal information, tells you more about research, tells you questions to ask when thinking about joining a research study. There are translations into Spanish as well, and a lot of good information at this site. There's also an excellent about 4-minute video on YouTube that is put out by the Office of Human Research Protection and about participating in research. So there's a lot of information available, and I think that people should be able to learn a lot by looking at some of these websites and just looking at the information. Let us know if you have any questions about any of this.
Thank you, Dr. Boshian. Thank you so much. There might be some in the chat at some point, but we haven't given the audience a chance to speak up for the interest of time. Next, we're going to play a video of Dr. Riccardi. I hope the volume's enough for you. I videotaped him on my cell phone and probably should have gotten a little closer up. For those of you who don't know, Dr. Riccardi's been studying neurofibromatosis, I think, since about 1969. And he's helped NF California since, I think I first met him in 2008 when he came to our beach event and was nice enough to do an Ask the Doctor one after that at Ask the Doctor meeting. And he's been a sounding board for Debbie and Maria and myself ever since. So here he is. Introducing Dr. Vic Riccardi.
Well, things started out a long time ago. About February of 1969, I made up my mind to go into neurofibromatosis. And about that same time, other people started to think about it, but nothing happened. And then I moved to Texas, and we really made it happen with the Baylor program. And now there have been so many research projects all over the place that you wonder which one should I be involved in. And I want to say that if we don't want only cancer research, we want research into the nature of NF1, We want to understand what the NF1 gene does. It does so much more than cancer. It involves your brain. It involves your bone. It involves your muscles. It involves your lungs. So when we think of NF1, it's part of our life, not part of cancer. And I urge people who have the disorder or family members of someone who has the disorder to consider research that seems that it might not have any payoff, but will give some vital information. And in particular, I want to say that I think that there needs to be focus more on what neurofibromin, the product of the NF1 gene, what neurofibromin does at the cell membrane. Right now we're working on something far removed from the cell membrane, the mech inhibitors. They're doing fine, they're helping a lot of people, but where NF1 really, that is the neurofibromin, makes its mark is at the cell membrane. So when there's a chance to participate in the cell membrane research, I've got to encourage you to go for that. And in addition, we used to talk about one gene, one phenotype. That is, if you know the gene, you're going to know the phenotype. Well, that's turned out to be a lot of nonsense. You can know the gene, but you don't know the phenotype. Not everyone with NF1 mutation has the same clinical problems. And so research that's sort of focused on understanding how factors other than the gene itself are contributing to the NF1 itself is really worthwhile, and I encourage the research for that. And to give you a couple of words, cell-cell cooperativity, how the cells work together to produce the problems or the peacefulness that you get. And another word for that is the epigenetics, the functions in addition to the genetics that impinge on the gene. That is not just that what does the gene do, but how does the gene do it? Right now, we're looking real hard at how the gene does it. So it's not a matter of looking at the gene itself. I encourage you very much to endorse and cooperate with research that goes that way.
Thank you. Thank you, Dr. Riccardi. Dr. Sarin, we're sorry to put you on the spot, but we'd like to know if you can possibly go next. We had a little snafu with Dr. Rosser.
Sure, absolutely no problem.
Okay, thank you.
Go ahead and load my. Okay. Set my timer. Sorry about that. Okay, great. Yeah, so for those who haven't met, my name is Kavita Sarin. I'm a dermatologist at Stanford University, and I'm on the Scientific Advisory Board for Inflection Therapeutics. And today I'd like to introduce you to Inflection Therapeutics, share with you the work that Inflection Therapeutics is doing to develop therapeutics for patients with NF1, I'll share the results of our phase 2A trial for a topical MEK inhibitor for cutaneous neurofibromas, which was completed last year, and share the design of our phase 2B trial for cutaneous neurofibromas, which is currently enrolling. So Inflection Therapeutics is a biopharmaceutical company that's focused on developing therapeutics to meet the needs of individuals with NF1. And it is led by an experienced management team, with deep industry experience in bringing drugs to market in both rare disease and dermatology. In addition to this management team, there is a scientific advisory board with expertise in both skin disease as well as NF1. So the focus of the current trials for Inflection are cutaneous neurofibromas. And as you know, cutaneous neurofibromas can appear anywhere on the body and occur in over 95% of patients with NF1. Although these can be both symptomatic as well as in some cases lead to social anxiety, there are few limited treatment options: surgical excision or laser therapy, which can be effective actually, but leave scars, and neurofibromas can recur after treatment. There are no FDA-approved therapeutics currently for cutaneous neurofibromas and no preventative treatments. MEK inhibitors are a very promising therapeutic for cutaneous neurofibromas, and this is because cutaneous neurofibromas arise due to biallelic or loss of NF1, leading to overactivation of the RAS pathway. And this leads to elevated phospho-ERK, which is downstream of the RAS pathway, and overactive cell growth. And MEK inhibitors block the RAS pathway downstream of NF1, inhibiting the RAS signaling pathway and, in theory, blocking cell growth. And the real potential of MEK inhibitors in neurofibromas was demonstrated in 2020 with the approval of cellumetnit for plexiform neurofibromas. And this showed a high efficacy of systemic MEK inhibitor cellumetnit in reducing the size of plexiform neurofibromas. And based on this trial, many have hypothesized that it would have a similar effect on cutaneous neurofibromas. But the problem with systemic MEK inhibitors is that they can cause side effects. And these side effects can include vomiting, diarrhea, rash, nail pain, as well as laboratory abnormalities. And for lesions like cutaneous neurofibromas, which may require prolonged treatment, it may be beneficial to have a safer medication that could be used for an extended period of time. So Inflection underwent a large campaign to develop a topical MEK inhibitor gel for cutaneous neurofibromas. And the discovery chemist screened over 100 compounds using a variety of assays to pick a compound that had 3 properties. It was permeable, so when applied to the skin, it could permeate the skin and get to the target neurofibroma. It was highly potent in blocking the RAS pathway when it hit the target neurofibroma, and it was also soft. And what it means by soft is that when it hits the bloodstream, it is rapidly metabolized so as to reduce systemic effects. And through this compound screen, the lead candidate, NFX-179, was identified. So last year, we completed a phase 2A clinical trial of NFX-179 in cutaneous neurofibromas. And this was a multi-center, randomized, double-blind, vehicle-controlled trial. 48 patients were enrolled with the diagnosis of NF1 and who had at least 6 neurofibromas that were treated during this trial. And these 48 patients were randomized to either low dose, medium dose, or high dose NFX-179. So there were 12 patients per group, and then 12 patients who were randomized to vehicle alone. And this was a small, short study of 28 days, which was intended to demonstrate whether or not NFX-179 could suppress the RAS-MAP kinase pathway in the tumors. So the primary endpoints were both safety of the topical gel, as well as the pharmacodynamic activity, the ability of this gel to suppress the RAS pathway in these tumors. So here are the demographics. So there were 48 subjects randomized, 47 completed treatment, 1 dropped out due to COVID-19. And the mean age was 47 years old. It was female predominant as well as Caucasian predominant. There were no significant treatment related adverse events reported. And the adverse events were not related to the study drug, such as sleep apnea or lower leg edema and so on. Interestingly, there was no acneiform rash, which has been seen with systemic mech inhibitors in this study. And high dose NFX-179 was able to suppress the RAS-MAP kinase pathway by about 50%, 46.9% here, and just generally had a dose-dependent suppression of phospho-ERK in the neurofibromas. What was unexpected was that we saw a change in tumor volume after only 28 days of treatment. And the reason we didn't expect this to occur in 28 days is the plexiform neurofibromas have a volume reduction on the order of 4 months. And we started to see, a 17% mean reduction in tumor volume in the 0.5% NFX-179 gel group as compared to vehicle. And these are just some pictures showing some flattening of the tumors, even at 28 days in the post-treatment as compared to pre-treatment photographs after treatment of 0.5% NFX-179. This is a responder analysis. So these are the percentage of tumors that had at least a 50% reduction from baseline. And we could see that 20% of the tumors treated in the high dose NFX-179 group had a greater than 50% reduction in volume. When we look at per subject, we again see a similar response with a 22% per subject mean response in the high dose group versus a 6% in the vehicle group. And I just wanted to remind, this is very early data because this is only 28 days of treatment for this study. One interesting thing we found was that in the high dose group, the response of the individual neurofibroma correlated with the suppression of the RAS-MAP kinase pathway, really, which suggests that the RAS-MAP kinase pathway is responsible, suppression of the RAS-MAP kinase pathway is responsible for the clinical response that's being seen. So the conclusion from the phase 2A was that NFX-179 gel is safe and well tolerated for up to 28 days. There were no treatment-related adverse events. There was no acneiform rash, which is actually commonly seen in oral MEK inhibitors, and had a very low frequency of local skin reactions, which were mild and transient and similar to placebo. The NFX-179 gel suppressed phospho-ERK or the RAS-MAP kinase pathway in the CNF tumors, meeting the primary endpoint. And despite that this was only a short 28-day treatment, there were early signs of tumor volume reduction. So now Inflection is currently conducting a phase 2B double-blind vehicle-controlled multi-center study. This is a larger study with treatment for a longer period of time. So the goal of the study is to enroll 150 adults with NF1 from over 20 sites across the United States. And each enrollee should have at least 10 cutaneous neurofibromas for treatment. The participants will be randomized to low dose, mid dose, or low dose, high dose, or vehicle. They will be treated for a total of 6 months. And the primary objective is to determine the effectiveness of low dose and high dose NFX-179 in suppressing tumor volume compared with vehicle gel. It will also be to determine the safety of prolonged 6-month treatment of NFX-179 on the skin. The clinical trial sites are scattered throughout the United States, but I just want to draw your attention to the 3 in California, in Fremont, which is the Bay Area of California, in Los Angeles, Dr. Roch's group in Los Angeles, and in San Diego. And you can see on the website a list of all of the trial sites across the country. And for those of you who have phones, you can also scan this QR code, which will provide you with more information on trial sites. You can ask questions and learn more about the study. And with that, I'll end. Thank you very much.
Oh, thank you, Dr. Sarin. And sorry about all the interruption, the beeps. We didn't realize texts were going to the computer, so we turned it off. It won't happen again. Thank you. Okay, so we're running a little bit ahead of schedule. Dr. Gia, are you prepared? Would you be able to speak now?
Yes.
Wonderful, thank you.
Okay, let me share my screen. Okay. Okay, good evening, everyone, and thank you all the organizers and everyone to participate in this. So our research is on the early detection and treatment of cutaneous neurofibromas. Hopefully our research can improve the quality of life of NF1 patients. My name is Wang from the Beckman Laser Institute and Medical Clinic. I believe not many of you know about this Beckman Laser Institute and Medical Clinic, so I give a really quick introduction. It's founded in 1982 with a gift from the Beckman Foundation. And the vision of our founding director, Dr. Michael Burns, is to build an interdisciplinary center which integrates basic science research, clinical research, and a medical laser clinic to offer patient care with vascular malformation and various skin disorders and skin cancers. So here is our research team. We have physicians specialized in laser surgery. Dr. Nelson is the medical director of our clinic. Dr. Kelly, the chair of UCI Derm. Both of them treat patients daily and actively participate in research. We also have scientists, engineers in the biomedical imaging and laser treatment. Dr. Milner is the director. He is supposed to come today, but on an airplane. And Dr. Tony, Dr. Sierra, and myself. I want to say a few words about Dr. Sierra. He is a retired, more than 10 years CTO at a major laser company. He has a family member with NF1. And he is the tireless driving force behind this research. So back to the cutaneous neurofibromas. Dr. Sarin gave a very good introduction. Is the most common tumor in NF1 patient. Appears around puberty, progressively, both the number and the size of the neurofibromas increase with age. Once it has increased to this size, the only treatment option is the surgical removal, which has significant permanent scar. Both the neurofibromas and scar are a significant burden to the patient. That's why we are working to develop a treatment option for really early stage or somewhere between the nascent and the flat. We call all of this early stage. As you know, the neurofibromas can appear on anywhere of the body, not necessarily one particular location. Therefore, we need a screening tool which can image a large area to find out where is the early stage neurofibromas. So this particular tool is called spatial frequency domain imaging, or SFDI. It's a wide field quantitative imaging tool which can give the skin optical property. Once we have a possible location of the early stage neurofibromas, we image with another tool called optical coherent tomography, which give high resolution structure, vasculature, and scattering of a small area, like 1 to 2 centimeter. We use one tool for screening, one tool for confirmation and validation, and to locate where is the cutaneous, early stage cutaneous neurofibromas, and then give laser treatment. This is the whole vision of this image-guided laser treatment of early stage CNF, especially for young adult. So I go to a little bit more technical details, but it's going to be not too much. So the first one is SFDI. It's camera-based instrument. Basically predict patterns on the skin. Then image with camera using mathematical models, we can tell the absorption in the skin and the scattering in the skin, which is basically the optical properties. This has already built in a portable, easy to use instrument. Pretty much anywhere of the body can be imaged with this instrument. So some results of the SFDI. Here is a upper left arm of an NF1 patient. You can see some visible neurofibromas and some area with a little bit discoloration. And the lower panel is the SFDI scattering map. I don't go to detail of this, but basically you can see the scattering of the neurofibromas is different as compared to the surrounding uninvolved skin. If we plot the scattering coefficient along different wavelengths, we get this curve. So scattering coefficient, wavelength for uninvolved skin, the black curve, and other visible neurofibromas or possibly early stage neurofibroma, the scattering is significantly lower. That's the result from this instrument called SFDI. So here is another result of SFDI is still forearm with a relative flat area and an area with significant visible bump on neurofibromas. SFDI gave more information. The neurofibromas is better with the better visibility in the SFDI. We also see quite some bumps in the seems normal area. You see the little bump there. What are those bumps? For the detailed structure, we need to use another instrument called optical coherent tomography. I think some of the physicians are familiar with this instrument. It's similar to ultrasound, basically deliver light onto the skin. Light bounce back, we measure the bounced back light and give the microstructures. So basically we put a probe, this is not exactly our probe, we put a probe on the skin and get the microstructures of the skin. So some of the result of the OCT, you see there are two visible, not too big, visible neurofibromas. On the OCT, we see the lower, here is the uninvolved skin. You see kind of the scattering, kind of uniform. For the CNF, we see significant lower scattering, like a dark area. We can predict the whole volume of measurement onto this attenuation prediction. And the neurofibroma certainly significantly higher visibility. We can also detect the vasculature surrounding the neurofibromas or in the neurofibromas. This is for visible CNF. So for early stage CNF, visible and here possibly an early stage CNF, I hope you can see it over Zoom. There's a little bit something there. So SFDI, lower scattering coefficient there. And this is the OCT image. We showed to dermatologists and other doctors, and a dermopathologist, they tell me, okay, this is not a normal skin structure. This is something abnormal. I can also show a video. You can see this thing change in look. It's unlikely a hair follicle. So we believe the OCT is able to further validate the SFDI results. So a quick summary of the imaging part. So far, we imaged 3 patients. I believe one of the patients is in the seminar. And SFDI shows optical scattering can be used to find where is the nascent early stage CNF, provide a map for screening. You know, we have the large body area like chest, back. We can use SFDI to screening for early stage CNF. And then the CNF can be analyzed as much higher resolution with OCT we can see how the tumor development, the development of the tumor, change with age. Will the early stage develop into a visible or not? And Dr. Sarin talked about the NF179 screen. We can use OCT to monitor in vivo without doing biopsy how the tumor changed with that cream. Okay, so that's the imaging part. Dr. Milner is supposed to talk about the laser treatment part. He's not available, so I give a quick introduction. The laser treatment. The basis of the laser treatment of different like Paul-Weinstein birthmark, hair removal, the basis is the theory of selective photosynthesis. It's a long word. I don't go into the detail. So I just show you a video. What's the meaning of selective photosynthesis? You saw two balloons, one inside, one outside. You may hear some sound from here. A laser will be fired. Oh, sorry. So here shows the outer balloon is still intact, but the inner balloon got popped. The reason is, the reason is that the inner balloon absorbed more light than the outer balloon. That's the one principle of selective. You need to find a target which absorbs light more. Yeah, here is another example. This is real treatment of spider wing. So with the delivery laser, you can see the target, which is the blood vessel, actually disappeared. This is the fundamental laser treatment Paul-Weinstein birthmark. Basically 40 years ago, there's no this kind of selective treatment. Paul-Weinstein, just like cryotherapy, even radiotherapy. So we hopefully this theory, this practice can be translated to neurofibromas treatment. So regarding the laser treatment of cutaneous neurofibromas, what's the target? What the target can selectively absorb laser? One possible target is vasculature. One study shows CNF has a lot more, 39 to 43 microvessels in the microscope field than uninvolved dermis. Dermis only 6. Therefore, we could do laser CNF treatment with a vascular targeting laser, which is commercially available for Paul-Weinstein treatment, like 595 nanometer laser, 532 nanometer laser. Dr. Sierra did a simulation how the laser treated cutaneous neurofibromas. It is certainly a simplified model. It has an epidermis layer, CNF layer, and dermis layer. With this laser applies 34.5 joule per cm square. You can see the red curve, basically temperature distribution in the neurofibromas layer could be increased above the damage threshold, but the surrounding skin is lower than the threshold. So basically only damages the neurofibromas. That's certainly a numerical simulation. This is the first target is vasculature. The second target is hair follicle. Study from other people showing their neurofibromas, the nascent stage neurofibromas here and here, often closely associated with hair follicles. The possible reason is the hair follicle is a source of stem cells, which could also be the source of neurofibromas. So what about we remove the hair with laser? Hair follicle laser removal is very mature technique and very safe. So we could perform CNF treatment, early stage CNF treatment with a hair targeting laser. So that's a very brief introduction about laser treatment of CNF. So right now we have an IRB approved clinical study for imaging and laser treatment. We need a patient, NF1 patient with or without visible CNF, not being treated for NF1 and associated conditions. 10 years or older for imaging, 18 years or older for laser treatment. So 2 visits, 12 months apart. Each visit take 45 to 60 minutes. Transportation can be reimbursed if necessary. You can contact our clinical coordinator or myself. I really appreciate the NF1 patient participating in the study, the NF network, NF California, and physicians who help, and the funding agencies. Thank you.
Thank you, Dr. Gia. That was very interesting. Dr. Leah, are you ready to go? It's a little bit early for you, but if you're ready, we'd love to hear what you have to say. Thank you. She muted.
Yeah, I think I can go ahead and go. I just had to. Okay, are you able to hear me okay?
Yes.
Yep.
Okay. Hello, everyone. I am Dr. Leah Yim-Fu. I am a neuro-oncologist at UCLA. I'm mostly see adult patients. So I'm also the clinical director for our UCLA NF and schwannomatosis program. And since I see mostly adult patients, I also see both NF1 and NF2 and schwannomatosis patients. We do tend to see more adult patients, and we work with Children's Hospital of Los Angeles when the patients become old enough and need more adult care. So in our team, we do have pediatric neuro-oncology as part of the team. They just don't tend to see as many patients. So most of our clinical trials tend to revolve more around NF2 schwannomatosis rather than NF1, although I did have an NF1 study of using the mech inhibitor with Neeratinib, and that was in clinical trial. The study has closed at this point. They have completed enrollment, so the trial is no longer open at UCLA or around the country, actually, because they are just wrapping up data at this point. They've already enrolled for completion of the trial. So right now we have 2 NF2 clinical trials that are open at UCLA. And one thing about our clinical trial is very similar to the NF1 concept. It's the same thing for NF2. All of the clinical trials for NF2 tend to be more molecular targeted agents. We know that with NF2, there is a mutation in the NF2 gene, and that protein interacts with a lot of different cellular pathway that lead to tumor growth and division and metabolism, as well as trying to get the tumor to create more DNA to grow more tumors. So most of the clinical trials around NF2 are using molecular targeted therapies that block the various pathways that interact with NF2. So currently the main trial we have is the Intuit NF2 Platform Trial. And that is a multi-institutional trial. I explain a little bit about what is a platform trial. It's really a novel trial design for NF2. This is the first of its kind for patients with NF2. A platform trial is a clinical trial where we actually are trying to assess and evaluate multiple drugs at the same time. So you heard about what a clinical trial is earlier on, and usually with clinical trial, we only test one drug at a time. However, in a platform setting, it is written so that we can actually test multiple drugs. We create something called master protocol that have common eligibility criteria so that specific patients meeting those criteria can go into the trial, and then they can be randomized to the different drugs that are on the platform. It allows for a faster way of developing treatments for a disease. So a lot of other cancers have platform trials, and brain cancer, which is something else I do as a neuro-oncologist, glioblastoma, we also have a platform trial there. And so us as a group of neuro-oncologists decided to adapt this type of trial design for NF2. The other thing about this trial is that it's also written so that there is an adaptive trial design. It's written in a way that a drug would be tested very quickly, and analysis are done early in the interim to decide whether it's even useful to continue on with the trial or not, instead of waiting until all the patients are enrolled before we get the results. So we would adapt the protocol to whatever drug that's working and drop whichever drug that is not working. So this is kind of the schematic of it. One thing about this trial also is there's also something called a basket trial. It allows different type of tumors. Any patients with NF2 disease with growing tumors, whether it's a vestibular schwannoma, a non-vestibular schwannoma, meningioma, or ependymoma, can go into the trial. So most clinical trial in the past may just focus on one type of tumor, one drug. In this case, patients can have growth, different patients with different growth of different tumors can go into the trial, and then they can randomize into different drugs. And we would try and adapt and move one drug that may be working faster into a longer trial to assess its efficacy. So right now the trial, because it's just started, we only had one drug so far, which is the Riccardi drug. And I will explain, this is all the trial sites that are available right now for the NF2, obviously in California. This is the location of UCLA. And then there's a couple of other locations, as you can see, mostly on the East Coast. We have been talking about trying to add in a little bit more in the middle because I feel a little lonesome by myself all the way out here. Like I said, it's any patients with a growing tumor that have these 4 different type of tumors. The tumors have to be large enough to be measurable, and the patients have to be at the age of 12 years of age or older. So again, NF2 is a condition that is mostly in teenagers and adults. So that's why there is that inclusion criteria. I'll explain a little bit about the Riccardi substudy because it's actually interesting so that you can get a sense of how the trial works. So the point of the trial is that we would just enroll 20 patients first and then do a quick interim analysis, making sure that there's at least one patient who had a radiographic response, that is, one of their tumors shrunk before we would move on and enroll more patients in stage 2. And also at this stage, like I said, at the beginning, there are 4 different type of tumors that patients with 4 different type of tumors that can be enrolled into this stage. However, at stage 2, we would just pick 2 tumors that seem to respond the best to the treatments and enroll only patients into that stage 2. And I must say we started this less than a year ago, and it's actually close to enrollment at this point because we enroll so quickly into the study. And we are working on a second drug to the trial, and that's called Neeratinib. And what Neeratinib is actually an EGFR inhibitor. Again, one of those proteins that interact with NF2 is an epidermal growth factor receptor, and is controlled by NF2. It leads to tumor growth. There has been other drugs similar to Neeratinib that has been in clinical trials for NF2 in the past that actually does show some tumor response in those trials. In mouse model, it looks like Neeratinib has even better effect than those other drugs. So we are very hopeful about bringing this into this study. We're also looking to see, can we predict how fast patients respond by using molecular biomarkers. So FDG PET scans, actually this part is only going to be done at UCLA, where we would image patients with a PET scan before they start the drug for a short amount of time, then re-image them. For the individual patient, we're not going to know yet whether it's helpful, but really the point is to learn, can we use these type of molecular markers as a quick way of predicting for future trials whether we can get early response. That way patients don't have to be on drugs for months and months and months before we know if it's working or not. So we're hoping that this arm of the trial would be ready. I'm hoping 3 months, but it's probably going to be around 6 months before it will be ready. The other trial that we have open now at UCLA is the Crizotinib clinical trial. That trial is part with the Neurofibromatosis Clinical Trial Consortium. And it's also just for patients with NF2, and it's only for patients with growing vestibular schwannomas. So this is a trial that is not a basket trial like the Intuit NF2 trial. Only if patients have vestibular schwannomas that are growing who can go into this clinical trial. It's a drug that, again, also in mouse model, found that when mice with schwannomas injected into them, that the drug can actually decrease tumor cell growth and lead to some shrinkage of the tumor. Again, patients have to have growing vestibular schwannoma. The trial at other centers actually do take patients under the age of 12, but again, since my center is really focused on adolescents and adults, we see patients over the age of 12 for this trial. And again, there has to be a somewhat of a size of a tumor with evidence of growth before the patient can go into the clinical trial. So this trial is probably going to be opening in the next month or so. So those are our current clinical trials. We are in the process of developing some new trial. Also, there will be a trial for patients with meningiomas that have NF2 mutation. So that will also include patients with NF2 disease with a drug called REC2282, which is an HDAC inhibitor. And to develop more trials for patients with schwannomatosis, and as well as finding partnership to do NF1 clinical trials that are focused on adult patients, and also in partnership with Children's Los Angeles. That's it. Thank you for allowing me to come on and kind of go over some of the clinical trials that are at UCLA right now.
That's wonderful. Thank you so much, Dr. Leah. Thank you. And sorry again for the interruptions. There's some sort of notification from the phone that's getting into the audio of this webinar. We don't intend for it to be your timer button or anything.
No problem.
Thank you. So next up is Herb Sarnoff, and he created Inflection Bioscience, and I'll let him tell you the rest. Herb, welcome.
Hi, can you guys hear me?
Yes.
Excellent. Give me 1 second. Is this on the screen?
Yes.
Yep.
Excellent. So, so my name is Herb Sarnoff. I'm the founder of a company called Inflection Bioscience. We're located in San Diego, California. Like probably a lot of people on the call, I am the parent of a child that has NF1. And several years ago, I was in transition in my career and decided to see what kind of a difference I could make with in the world of NF. I had befriended Dr. Riccardi and gotten a lot of inspiration from his teachings and his approach to NF, and tried to carry that through. Here's some examples of things that have happened in our life with scoliosis and tumors and bone tumors, etc. So today I'm going to kind of share with you a little bit about how I kind of perceive the world of NF1 and the current unmet need, and the fact that it's an orphan drug and what that opportunity presents. I'll share a little bit about the company and where how we're approaching NF1, which is kind of different and along the lines of what Dr. Riccardi was talking about, kind of targeting the underlying protein. I'll tell you about our approach and a little bit about some proof of concept or early proof of concept that we have. So you'll see here that I'm nowhere near any of the clinical trials that are being discussed today. We've got several years for that to be in that position. I'll share a little bit about our team and our goals moving forward. So NF1, as most people here know, is a rare disease, 1 in 3,000 people. So that's 2.5 million people worldwide with NF1 and 125,000 people in the US. So while there's a very large population, it is an orphan disease. And the government gives a lot of benefits to companies that are targeting and going after orphan drugs. So it's a strong unmet need, but there's a big opportunity to make a difference and for a company to make a difference. Here are a lot of the symptoms of NF1. People know these symptoms. It affects many, many different body symptoms or body systems, from tumors to bone issues to cognitive issues. You'll see here in a second what we're doing at Infixion. Well, so I'll say also there's one treatment available today. So Koselugo, most people know, is available. It's a treatment on the market today. It's got a very high list price. You'll see that there. Its current list price is $150,000 a year for this drug. And it does provide tumor shrinkage for a subset of the NF population, but there are a lot of side effects with the drug. So it's not necessarily an ideal situation, but it is providing benefit to people today. So what are we doing at Infixion? If you think about a cell of a person that does not have NF1, both sides of your gene produce protein called neurofibromin, and it produces, in the end, a total of 100% of what you need to have that cell be normal. But for somebody who has NF1, you could think of there's a mutation on just one side of the gene, and that means what you have is something called an autosomal dominant genetic disorder. When the symptoms appear, even though you have the mutation only on one side, that's autosomal dominance. In this condition, you also have a situation where you have a lot less of the good protein. So in this case, the nominal case, you have 50% good protein in that cell, and the other 50% is a mutated protein that's non-functional. So what are we doing at Infixion is we're trying to treat this at the cellular level by attacking the production of NF1 protein, neurofibromin, the modulation of neurofibromin and how it's controlled inside the cell, and then the actual storage of protein itself and how it degrades over time. So that protein goes away and you make new protein. And we think that if we can attack these different areas within the cell, that we can have a major difference in terms of how the body functions. So why is neurofibromin so important? Dr. Riccardi touched on this. Neurofibromin is kind of at the center of the issues associated with NF1 and the different symptoms. And you can see that NF1 sits and affects this different pathways called the RAS pathway. There's the CAMP and CRMP2 pathways in the brain. And NF1 impacts all of those. And you can see where Koselugo is targeting kind of downstream here in this mech pathway. And our goal is to kind of target upstream and target right at the root cause of the problem, which is this lack of neurofibromin. So we currently have 3 different programs looking at these different elements of NF1. I left some of the technical terms in here for the doctors on the call, but we're looking at transcriptional regulation of NF1, which is essentially making more of it in the beginning. We're looking at protein degradation, so we want to keep that protein around longer. And then we're looking at something called microRNAs, which is actually a substance that kind of puts a brake on the production of neurofibromin, and we want to remove that brake. And we currently have NIH grants associated with each one of these 3. So we've gotten 3 NIH phase 1 SBIR grants. So we're off working in these 3 different areas. So some of the accomplishments, we've built a really solid execution team. I'll show that in a second, and the advisory board. We have these foundational grants. We have a lab infrastructure here. I'm here in the lab today. And we've written a white paper that's soon to be published on the rationale for why neurofibromin is really, and haploinsufficiency is really a key factor. We've also built a number of tools. We've built some reporter assays, which allows you to screen for drugs. We've built a new monoclonal antibody for NF1, which allows us to more accurately measure neurofibromin inside cells. And we've built some new cell lines, which help us to control how we test and look at neurofibromin in the lab environment. So we're still working at the cellular level, where we haven't even graduated yet to the animal studies. But it's kind of a funnel. So we were starting today with a known drug. So we're looking at FDA approved drugs. We're looking at a large repurposing library, which is a library of drugs that have been in clinical trials for other things, but were, for whatever reason, not approved. And we're looking, but we know that there's a safety, we have kind of safety knowledge about each one of those drugs. We're trying to find potential drugs from this repurposing world. But then you funnel it down. You try to confirm your hits and see if you can increase NF protein, and then see if you can affect some of those downstream pathways that we talked about in the earlier slide, and then compare it on a safety level to kind of ideal drugs, and then do some in vivo testing in animals before getting into clinical trials. So we're at the early stage. So this is a proof of concept. So we took the FDA library and we screened it using one of our reporter assays. And we found this drug, we're calling it the INF55. And you can see as in a dose-dependent manner, we can see an increase of NF1 protein when you give cells this drug. So we can see a 75% increase in neurofibromin production. This drug doesn't have a great safety profile, so this is not the drug that we're going to go to market with, but it is proof of principle that we can treat cells, see an increase in NF1 protein, and we're currently working to show how that increase in neurofibromin protein is impacting the downstream pathways of NF1. So this is our team. So it's a small team, myself and a PI here in San Diego, a couple of advisors that are day-to-day working with us on strategy and approach. A pretty world-renowned scientific advisory board. So Dr. Korf and Descaccio and Dr. Yohei are well-renowned people in the NF space, and a number of collaborations that we've developed with University of Wisconsin and Frederick National Lab, Scripps Research here in San Diego. So we're starting to kind of make progress on becoming really a player in this community. So where are we targeting? When you're fixing the underlying root cause of the problem, you couldn't think that we might impact any one of the different NF1 symptoms. We could improve tumors, we could improve cognitive issues, we could improve bone issues. But when you're running a clinical trial and when you're running animal studies, you got to decide early on what to focus on. And we went through a process with the NIH called ICOR. And during that process, we interviewed over 100 people in the NF1 community, so, patients, parents, NF clinic directors, NF1 specialists. And through that process, we came to the conclusion that the cognitive learning and social deficits in NF1 are the thing that is impacting the most kids, the most people early on in life. That if we're going to treat people early, this is the symptom that would be most interested in by the community to treat. It's also the thing that is most dependent on this lack of neurofibromin protein. We think that it's strictly the neurofibromin protein levels that are causing these cognitive learning and social deficits. So we made the decision that this is where we're going to target, and this is where we're going to run our clinical studies and our animal studies. So big picture, because we're focusing on what the body is doing right, which is making good protein on this side of the gene, that's the good side of the gene. It doesn't matter what mutation you have. So we've already identified over 4,000 unique mutations in NF1, but this treatment protocol, this treatment path doesn't depend on what your mutation is. We're focusing on what your body is already doing right and trying to increase the production of protein from that good gene. And because we're focusing on the root cause of the problem, we have at least the potential of impacting all of the different NF1 manifestations. So we believe that we can both prevent and treat manifestations with this process. And we're also avoiding some of the, there's people talk about gene therapy and the benefits of gene therapy, which essentially be where you would go in and fix this side of the gene. You would fix the mutation, fix the problem on this side. But there's a lot of challenges because of this large number of mutations and the very large size of the protein itself, and the fact that it needs to get into so many different cell types in the body. This approach of gene therapy has a lot of challenges moving forward. So we believe that a more traditional path of drug discovery using what's called small molecule technology, which is what 98% of all FDA drugs today are small molecules. We believe that's the right approach for NF1 in the near term, and that's where we're focusing our efforts. So that's Inflection Bioscience quickly, and I'm open to answer a couple of questions. I think I have a couple of minutes left. They said I could take 15 instead of 10. If there's any questions or if you want to move on.
Okay.
Herb, thank you so much. We're not set up for questions tonight.
Okay.
But we really appreciate your presentation, and it's exciting to see your progress. Thank you. Welcome. Next up is Dr. Cynthia Campen. Dr. Campen, thank you for being
Hi,
here.
everybody. Debbie and Katie, thank you so much for organizing this. You guys continue to amaze me in all that you do, and this community is really lucky to have you. And thanks for inviting me. Herb, do you mind stopping your screen share so that I can share my slides?
I was trying to find it. There we go.
Awesome. And Herb, you are so inspirational with all that you've done and how far you've taken this work, truly.
Thank you, Dr. Campen.
It's so fun to watch your presentation and to see how far you've come already. So I look forward to seeing more from you guys in the future. I'm just going to get myself scheduled all together here. So, and I'm a little late tonight. It was open house at my kids' school, so my apologies. I missed a lot of great presentations, I know. But let me tell you what we've got at Stanford Children's for folks with NF. I am one of the pediatric neuro-oncologists there. I'm a child neurologist as well. Neurocognition in NF is very near and dear to my heart, as Herb was saying. It does affect cognitive issues and learning challenges, as you all know, affect upwards of 70% of kids with NF1. And so what we're trying to do is identify neuroimaging biomarkers that can predict which children are going to go on to have learning challenges. And the hope is that we can select a population of folks that would be good to intervene on for someone like Herb and other studies that are happening in terms of therapeutics for neurocognition in NF. And so currently we are just trying to get a lay of the land with what does the NF brain look like in terms of white matter, which is responsible for much of the dysfunction that we see. And so we're taking all comers, folks with NF from ages 3 to 18, and doing an MRI and about a half an hour of neurocognitive assessments on a computer. It's kind of fun, like computer games. And so we are have that open and running, and just give us a call if you're interested in participating. In addition, in terms, so we have looked at our retrospective data, looking at diffusion weighted MRI, which is a way to track the white matter tracks along the brain. And this is just a representation of one of our actual patients' white matter tracks in the brain. And we're able to pick out the different tracks in the brain and look to see the actual integrity of each of those tracks. And we've identified that there are really significant microstructural differences in the NF brain, that younger children have more significant differences than older children or adults. And we, as I demonstrated, we have a prospective study open to look for specific age-related differences. And then in addition, we have some tumor-specific clinical and observational trials available at Stanford Children's. We are running the SpringWorks Neeratinib study for plexiform neurofibromas, but that is closed to enrollment. We also have pediatric brain tumor consortium studies open, one of which is specific to NF1-associated recurrent or progressive gliomas and optic pathway or otherwise. And that's looking at the use of trametinib with hydroxychloroquine to synergistically affect better tumor control. We also have the Children's Oncology Group clinical trial open for newly diagnosed or previously untreated NF1 associated glioma. And that's looking to randomize between cellumetnib and carboplatin vincristine, which has to date been standard of care for gliomas. We also have the NF-associated optic pathway glioma natural history study open that's trying to determine which children with OPGs are going to need treatment moving forward because it's so difficult to predict. And then coming soon, probably in the fall, we are going to be opening the Parexel NF2 meningioma clinical trial using an oral agent. And we're just going through all the red tape to get that open now. If you're interested in talking to us more about the clinical trials we have open, please go ahead and have your child's physician call our Stanford Children's referral number, and they can request an NF referral for our clinic in neuro-oncology. Thank you so much for your time. I appreciate you guys. I will stop sharing.
Thank you, Dr. Campen. That's wonderful. And next is Dr. Baca. Thank you for joining us from far away. We have your brochure if you need it, if you need us to share it. Do you need us or can you share?
Let me see. Can you hear me?
Yes.
Okay. Let's see. Why don't I just have you share it because I'm
okay.
Trying to get it. And it's not opening.
Is it showing correctly?
Most of it, there's a little overlay on it. There, now it's perfect.
Okay.
So thank you very much for having me. I'm Dr. Nicole Baca. I'm one of the pediatric oncologists at Cedars-Sinai. And we have an institutional study that we wanted to share with the group tonight that we are actively recruiting for. It just was officially opened 10 days ago, maybe. So it is an multidisciplinary study that we wrote out of Cedars, and I'm the PI on this study. So I'm working with our pediatric geneticists, and there's actually a medical oncologist that's on the study, as well as our biomedical imaging research institute called Berry, based out of Cedars. And we were brainstorming actually in the midst of COVID, trying to think of, are there any good study ideas that we had for this kind of a population? Because our Berry Institute has been able to, in adult patients, use MRI imaging paired with artificial intelligence to predict cancer in high-risk patients before it happens. So they've had studies that they've done with prostate cancer and with prostate, and it was pancreatic cancers, that they were able to predict about 2 years in advance before the cancer developed. And we thought this would be a really great thing if we could use it in children with NF1. So particularly we wanted to look at two things, and I know this population is going to know a lot about NF1, so I wanted to go into too much detail, but the plexiform neuromas, can we predict who's going to have higher burdens of those? And although it's extremely rare, can we predict who is going to have a plexiform neuroma that transforms into a malignant peripheral nerve sheath tumor? And so we wrote this study to try to look at a couple things. First, we want to use these MRI techniques paired with artificial intelligence in children. It's never been done before at Cedars. So the first part of the study is going to be taking kids from 5 to 17 years of age who can have an MRI, and it's a whole body MRI from head to knee. It'll take about 70 minutes. And it's not as detailed as if you go to get an MRI because you felt a plexiform on the arm. It's a screening MRI, so a little less detailed, but we can get a lot covered in a very short amount of time. And we want to see if we can use, if kids can handle that 70 minutes without sedation. And we want to see if it's reproducible, meaning we do it once and then we do it again a month later where we don't expect to see changes, and make sure that it's the same, basically, which is what we would expect because it's been done in adults. And for the children that do the study in adolescents, we would get a read from a radiologist on one of those. So if we see anything that's a little bit concerning, either my team at Cedars or your primary team can go ahead and order a more detailed MRI to get more of the details of the area, a clinical MRI. And then about a year later, so 9 to 15 months later, we want to repeat that MRI and we want to see if there's any change. And using those images along with the other images that Cedars has just de-identified that have markers on them, like this patient had NF or this patient had a plexiform neuroma. They've de-identified that and they're going to retrospectively look at those for all comers in the hospital, see if we can find something using artificial intelligence that helps us predict who's going to have higher burden of disease. And if that works, maybe we'll be able to use this in the future to help figure out maybe who needs intervention sooner rather than later. So the study is open and we're recruiting young children and adolescents. So if you are interested in doing one of these MRIs, we have some compensation because it is a lot of time and it's 3 MRIs, and we would give you some information from a radiologist, but that's our study. So it's open to anyone who'd like to come and over to Cedars and have that done. And then my phone number or my contact information is there, and we can give you more information as needed.
Wonderful. Thank you, Dr. Baca. We really appreciate it. And next up, we have Dr. Ruben Antony from UC Davis.
Hi, can you hear me and see me?
Yes.
Thank you. Let me see if I can share my slides. I have just a few slides. Let me see if I can share them. So as mentioned, I'm Ruben Antony. I am one of the pediatric oncologists at UC Davis Cancer Center in Sacramento. We have the NF1 clinic, or the NF1 program. As a part of the NF1 program, we take care of both children and adults. I mean, I myself and my nurse coordinator colleague are both completely pediatric trained, but we were told mainly by our colleagues at the Mind Institute here at UC Davis that there was a need for there was a kind of a need for a structured service for adult patients with NF1, and so we've just started seeing adult patients with NF1 as well, which is interesting in many ways because I'm completely pediatric trained. So we certainly don't offer the adults lollipops, I can tell you that. So we've just completed the Springwork study that I think had several participating centers here in the US and in California. The study went quite well. We recruited several patients, and within the little snapshot that we have, I think our patients did benefit from participating in the study. So I have just a few slides to tell you about a study that we are setting up right now. This is a study that it's the FCN 159 study, and it's sponsored by a drug company called Shanghai Fosun Pharmaceutical Industrial Development Corporation. I think it's an international multi-center study that has 2 or 3 centers within the US. We've been approached to participate, and we are hopefully going to be able to actually join the study. The negotiations with the company are ongoing. So the study is a multi-center open label study. It has a phase 1 and a phase 2 components to it. There's some preclinical and some preclinical. There's some preclinical data, and there is some data mainly in treating melanoma patients, which seem to support the efficacy of this molecule in shutting down the RAS map pathway. So there are 2 phases, like I just said. We are hoping to have the study open by July of this year, or hopefully at the latest by August. Phase 1 is open to patients older than 2 years. There are 2 cohorts, adult and pediatric, 60 participants in total in the phase 1 part of the study. And as with any phase 1, it's determined safety and tolerability and to inform dosing for phase 2. The phase 2 is open to patients older than 2 years. Again, I think till the age of 70. Adult and pediatric cohorts, 100 participants in total, and to assess pharmacokinetics, adverse effects, and efficacy. Just a few more details about the study, and then I'll stop. The medicine or the trial drug comes as a tablet. We are in discussion with the sponsor as to how the tablet's going to be administered to very young children. That's something that we pointed out to the drug company. It's administered once a day, and once a day daily for every day of a 28-day cycle. The duration of the study is 2 years. The drug is stopped if the sentinel plexiform that's being monitored progresses. The trial inclusion criteria are fairly standard, so I'm not going to go over all of them. I'm just going to mention a couple of them. Clinical or molecular diagnosis of NF1, an inoperable or troublesome plexiform, and a measurable lesion. In terms of exclusion criteria, really, again, fairly standard. The one that I think could be an impediment to patients participating is that any treatment at any time with a mech inhibitor is an exclusion criterion. I'm not sure why that is, but that's the way they want to do it. The other exclusion criteria are just chemotherapy in the last 3 months, any targeted therapy or biological therapy in the last 14 days, blood cell growth factor 7 days, radiotherapy 4 weeks, participation in another clinical trial 4 weeks, and any history of a malignant tumor that's NF1 related or unrelated. So we should have the trial up and running in July or August, at which point we can start to recruit patients. Thank you. I think that's it from me. Thanks.
Thank you, Dr. Antony. Next is Maria Powell, our third of the threesome of us that's NF California. She lives in the middle of the state, so we work all 3 of us together. And Maria has a few things to say before we conclude. Thank you.
Okay. First of all, thank you so much for attending tonight. Both the presenters and the attendees were really grateful that we're able to have this type of event. We never would have expected to go in this direction. We spent lots of hours and hours traveling, hosting events at different campuses and different medical facilities, and that's what we enjoyed doing. And now that isn't necessarily a possibility, but we're excited at how available this becomes for us. So where Debbie, Katie, and I live in 3 different locations, we do like to travel, but this makes it available on a Tuesday evening for everyone, instead of just those who can afford to travel to San Diego or to Sacramento. And so we're very grateful for the technology. Now, we do have a big question. I would hope in the chat you could just quickly just mention how you found out about this webinar, because we'd like to know what the best means of reaching out to people is and to finding you. So if you could just quickly, whether it was the email, was it Facebook, was it through National NF or National NF Network, or word of mouth, or maybe from a doctor, just that would be great. While I'm talking, if you could add that, thank you. So it was fun to watch the chat. As people were saying hi to each other and greeting each other, we miss each other, and we do miss having those face-to-face events. So I'd like to advertise and promote our next face-to-face event that we have currently scheduled is our beach day. And it's one that we've traveled from all over California to be able to go, and it's been wonderful. And so we're excited to be able to have that again. And the date on that is July 16th. So keep in mind, mid-July, if you have the ability to go down to Huntington Beach, that would be wonderful. Also, we're curious, if you would like to host an activity in your area, please let us know. because we want to have all of California, not just in the towns that Debbie, Katie, and I live. I don't think any of you want to drive to Chowchilla, California anymore for the events that we have had here. You know, we're kind of in the middle, but we're middle of nowhere. So please let us know where you would like to have an event. They're laughing at me because they've driven to my hometown more than they would wish. So if you would like to host an event, email info@nfcalifornia.org. And that way we can know to reach out to you and hopefully work to better represent all of California, because that definitely is our goal. All three of us do this because we're passionate about it. Our sons brought us to this diagnosis, and we're very grateful for each other, that we have each other, and we want that friendship to link all of us. So we're very, very excited to start having face-to-face events. And if we want to have more virtual events where we can just get together and chat, let us know that too, because we want to know what kind of events that you would like to have. And Debbie and Katie, anything else you wanted to add?
I was just going to add that Carter wants to do a cornhole tournament. So he's been begging for a month. So today I told him if he gets his driver's license by the end of summer, he's 20. Mom wants to stop being the chauffeur or the Uber driver.
Okay, so let's plan to head to Northern
by the end of August. If anyone wants to, whatever, have any tips, but that's that's his plan. So hopefully you guys will come to Dixon or wherever we have it.
Okay. And as you notice, we try to take our kids' and our passions and work with that. And so if there's something you're interested in teaching us or showing us or somewhere you want to take us, let us know.
Yes. And again, we're super grateful for all this, for all the presenters and how you stayed and listened to each other's presentations. I feel like that is one of the gifts that Zoom has given us, for people to be able to know who else is out there doing this work, and hope that you'll make some connections as well.
I just have a quick question, if that's all right.
Sure.
Is that okay? So I missed the first part of this discussion, and I'm sorry about that. But another drug company kind of at some point in the last year approached us to be a potential study center for a cutaneous preparation for cutaneous neurofibromas, and which we were quite excited about, because we really feel that's a need for our patient group. Then for reasons not clearly known to us, the company dropped us. They just told us that we're no longer in consideration. Is there any center that in Northern California that has or expects to have this trial opened up? It's using a kind of a topical gel on cutaneous neurofibromas as a form of treatment.
This is Kavita. I think if this is inflection, it's the trials. There's a trial site in Fremont, which is the closest one in the Bay Area, Northern California.
And which hospital would that be?
That's Dr. Dewan's site. It's a clinical research organization.
I see. Okay, thank you.
If everything went well, this presentation, the Zoom meeting was recorded and will be up on NF Network's website under webinars, and you could go through the presentation that was done on that inflection, and then see a little bit more about the study sites and everything.
And the webinar that we did last May too is on there, as far as facilities throughout or
NF Care in California. What other places besides, say, UC Davis, who else is seeing patients in California, which could be helpful for clinicians. So
Thank you.
That's it for us. Thank you all so much.
Go enjoy your evening. Thanks. Bye.
Bye.
Thank you.